An AGI needs to be able to answer simple questions. Complex questions too, but sometimes the simple questions are more difficult. This video starts by pursuing a few that are so obvious any child can answer them. And while we have systems which exceed human abilities in narrow areas, I contend that answering the simple questions is fundamental to general intelligence, and that the easy questions are not so easy when you try to answer them in an AGI sort of way. Consider this picture. If we say that A is red and that B is red, then ask, what color is C? A child can answer that C is red too. I could easily write a program which could answer this question too, but it doesn't make much progress towards AGI. The key is to see how this simple question is representative of an entire class of similar questions. Let's consider a slightly more difficult question. I'll add that C is lighter than B. Now, is A lighter than B? This second question adds the concept of relationships. In my videos on the Universal Knowledge Store, I showed how neurons can store relationships, but consider that information can have many relationships. Light or darker, nearer further, bigger smaller, sooner later, before after, and on and on. I can also encode any number of relationships, but can an AGI infer its own new relationships the way a child can? So while I'm asking simple questions about a few colored squares, I'm really addressing an issue which is fundamental to AGI. As I begin to implement a system which can address these simple questions, it becomes obvious that the answers are too complex for a single video. So in this one, I'll just talk about how color and other information can be represented, while in future videos, I'll dive into the relationships. I'm going to start with a biologically plausible model of color representation in the brain. An AGI need not follow this model, and it might be considerably more efficient to take a different approach. But this approach illustrates some constraints and shows how color perception could be directly equivalent to hearing or any other sense. To provide some test data in the brain simulator, I've created a module which can loosely represent a single pixel of the retina. You select a color and the module sets the firing rates for red, green, and blue neurons, each with a small amount of random noise thrown in. A lot more processing goes on in the retina, but we'll just focus on this one aspect. In general, any sensory nerve fires faster to indicate a more intense signal, just as these retinal cells fire faster with greater color intensity. If we look at the spiking of these three neurons, we can see that the red neuron is firing for this red pixel. But what if the pixel were orange? Or this slightly different orange? You can see that determining which color is which is a bit more difficult. It's not at all obvious that specific firing patterns represent one color or another. The primary difficulty is that the signals are not synchronized. If you look at the spikes for any short time frame, there are many different spike patterns representing the same color. Another constraint is that because of the neuron's refractory period, an individual neuron cannot fire any faster than 250 Hz, or once every 4 milliseconds. At the same time, based on the speed you can see or hear things, whatever information the signal represents must be conveyed in less than a 30th of a second. So our red neuron could be firing once every 4 milliseconds, once every 5 milliseconds, or so on. And how can other neurons detect this difference? At some point, your brain needs to be able to differentiate colors into individual spikes, if only to be able to say the words you've associated with the colors. This means that color signals cannot just cascade through the multiple layers of recognition. At some point, they must be decoded. Focusing still on the red neuron, there are two ways to detect the firing rate of any individual signal. The first is to define a specific time period and count the number of spikes in that period, and the second is to measure the interspike timing between adjacent spikes. The remainder of this video will show how these two functions are implemented in neurons. Starting with the first approach, let's take the signal from our red neuron and put it into a series of neurons, each firing the next. Think of each one firing 1 millisecond after the previous. Then you add synapses with weights adjusted to the various spike counts. You can do this all by hand, but I wrote a module which does it automatically. The number of levels which are detected and the time frame can be adjusted by changing the dimensions of the module. For details, have a look at the Brain Simulator's chain counter module. Remember that you'll have to replicate this process for red, green, and blue, and repeat the entire ensemble for each pixel in the visual field where you want to recognize color. You can observe two things from this module. First, the more different levels you want to detect, the slower the process will be because you need a longer chain. Also, the more different levels you want to detect, the smaller the differences in the synapse weights, and this will lead to noise issues. These limit the number of distinct colors you can detect and the speed with which you can detect them. This brings us to the second approach, looking at the interspike timing. This solution uses a neuron's inherent leakage. If, for example, you stimulate a neuron with a weight of 0.75, it will not fire, but its internal charge will initially be set to 0.75, and if a second identical spike is received before the internal charge has decayed below 0.25, the neuron will fire. Otherwise, it will not. With an array of neurons with different synapse weights or different leak rates, you can detect the timing between any two adjacent spikes. For this solution, I also created a module so that you can adjust the number of detected levels by changing the size of the module. And it has a column of short-term memory, so it's easier to see the single spikes of recognition. This solution only detects the signal timing of every other spike interval. You can make this solution twice as fast by adding another column of neurons to detect every spike interval instead, like this. For details, see the Brain Simulator Rate Decoder and Rate Decoder 2 modules. Measuring interspike timing has significant advantages over counting the spikes in a fixed period. It is faster and fewer neurons fire, so less energy is consumed. Measuring interspike timing also doesn't rely on the speed of the neurons. In fact, this circuit can recognize differences in spike timings to any arbitrary degree of resolution, limited only by noise and the consistency of the synapse weights and linkage. In the computer, all of this could be done numerically far faster than any of these approaches, but these approaches show some of the constraints your brain works with. In coming videos, I'll show how to combine these signals to learn any color and how this information can be used to infer relationships. Be sure to like and subscribe to this channel to be notified for new videos, and share with your friends. And as always, thanks for watching.